Okay, I think Catherine Howard, the moderator, has momentarily disappeared from the meeting. Yeah, I think she has some problems with her computer. But so I'll make a start with the first presentation. My name is Leif Skot, and I was head of forage plant breeding at Aberystwyth University until the end of last year, where I retired. So I now have an emeritus position. So I will talk about the genotyping platforms that are especially relevant for the eucleg project. All genotyping nowadays start with single nucleotide polymorphisms as the basis for all genotyping, and as you can see here. So it's either a change in base at certain positions, or it can also be insertions of some sequences or deletions of them. So everything we do is nowadays is based on polymorphisms in the basis of the genome. And so why are we interested in genotyping in crops? So for the first, it's to do with identifying genetic diversity in germplasm. You've already heard about that yesterday. So because genetic variation is the basis for breeding, without that, you might as well give up and go home. And you can measure or you can get sources of novel variation that you need to improve your breeding program or introduce new traits. And it'll also important in terms of improving varieties' resilience to climate change. And of course, pest and disease resistance is important as always. It may also, I'm not an expert in heterosis or anything, but it could be used or in determining which parents to use to improve or to get heterosis, and also identify parents that may give transgressive segregation, so traits that are sort of beyond what was there in the parents. The other reason why we're interested in genotyping is GWAS studies, or genome-wide association studies, and that was talked about quite a lot yesterday in the 3 crops that were treated there. And you want to associate genetic markers with phenotype, you want to identify QTL, and it'll also give you a possibility of diving into the genetic basis of a certain phenotype that you're interested in, and identify markers for breeding. Marker-assisted selection and so on. Finally, another thing that was mentioned quite a lot yesterday and shown examples of is genomic selection, where you identify breeding values of genotypes or populations based on all the marker effects of all that you have identified throughout the genome. So you can then predict the best candidates for crossing based on the genomically estimated breeding value, and thereby try to shorten the breeding cycle and thereby save time and money in the breeding program. And the final reason I wanted to mention is, of course, the price, which I'm sure many of you have seen this slide before, where since 2007, when next generation sequencing was introduced and really started to gain in popularity. Ever since then, there's been a dramatic drop until you reach sort of $1,000 per genome for the human genome. It seems to have flattened out since then, but maybe that's just because this was the goal that people were after. So I think there's maybe prices will start dropping again. So it's certainly a dramatic change of possibilities for genotyping of all sorts of crops. So the current genotyping platforms I'm going to mention is whole genome resequencing. Although we didn't do that in the eucleg project, I feel I ought to mention it. And then there are also SNP arrays, of which you have seen examples of 3 crops, of the 3 crops yesterday, and then reduced representation sequencing, which is what was used for genotyping in red clover and alfalfa, which you will see examples of later today. So some of the considerations for which genotyping platform to use are listed here. For example, linkage disequilibrium. The phrase was mentioned quite a lot yesterday, and this is quite important because it determines how many markers you are going to need. And linkage disequilibrium, it is quite often linked quite closely to whether you have an autogamous, orthogamous, or allogamous, or inbreeding or outbreeding crop. So we have examples of all the 2 types in the 5 crops that we have working with here in the eucleg project. And then, of course, as I said, price per sample is very important for everybody. And finally, ploidy, as many of the important crops that we are working with are polyploid, and this changes or makes things a little bit more complicated usually. So I think I'll sort of go back to basics a little bit. I wasn't sure what audience I was presenting to, but I decided to include a little bit of what linkage disequilibrium actually is. So on the left, you can see an example of complete disequilibrium. You have a number of genomes illustrated there, and there are 2 loci where there is polymorphism. And if you look at locus A, there's polymorphisms on between A and G, and locus B, C, and T. But they're all, no matter which of the haplotypes or which of the genomes you choose that have A at locus A, you will also have C at locus B. So the probability of finding C at the locus B, provided you have A at locus A, is 1. And on the right-hand side, you will see an example of complete equilibrium, where there's a 50-50 chance of finding a T or a C when given that you have A at locus A. And likewise, if you have G at locus A, there's a 50-50 chance. So the 2 loci are independently segregating. So and the sort of the basic measure of linkage disequilibrium is at the top there, which is the probability of finding A and B together minus the multiplication of the probabilities of each of the 2 allele frequencies. So if they were independent, so it's the product of the 2. But that measure depends on allele frequencies. So there was a wish to normalize the expression, so it varied between 0 and 1. So that resulted in the equation at the bottom, which is the one that are almost exclusively used, or certainly in the presentations here in eucleg. So which is the sort of the correlation between the LD and each of the 4 possible allele frequencies. So if you take an example here, where you have partial disequilibrium between 2 loci. And so let's say that marker 1 there is a QTL that explains 10% of the total phenotype. So it's not it may not be the causative SNP, or so SNP marker 2, which is the one maybe you have in your genotype platform, that explains 25%. So the linkage disequilibrium is 0.25, but it only explains 2.5% of the total phenotypic value, because that's only explained by 10% at locus 1. So it requires quite a large sample size to gain sufficient power or a higher value of linkage disequilibrium. So that's possibly why there's been so many attempts that have not succeeded in finding sort of single markers that can be used in marker-assisted selection for some of the complex traits that are really important in breeding programs, because it requires a really high density of markers. Anyway, I will now go on to polyploidy, which is also an important factor when you choose a genotyping platform. There are 2 types, allopolyploidy, and that is the combination of genomes from 2 related species, and through some sort of hybridization and subsequent chromosome doubling. And so examples of that are bread wheat and white clover. They sort of behave like diploids when it comes to assortment of the chromatids in the the other one is autopolyploidy, and that's basically when there's a chromosome doubling going on as a result of failure of meiosis or mitosis, and examples of that are potato and alfalfa. And paleoploids are formed a long time ago, and sort of our ancient polyploid, and they have diploidized over the millennia, and examples of that are maize and soybean. So the way in which it impacts on genotyping is, there's how do we tell the difference between allelic SNPs and homologous SNPs on the other genomes in the crop, and there is also that results in the competition for primers when you talk about allele-specific primer methodology, as you do in array technology. So in next generation technology, such as the reduced representation sequencing, as I'll talk about shortly, you need a larger coverage compared to these are numbers I've taken from the paper quoted at the bottom there. Sort of they mentioned 7.7 times coverage, which is the number of times a certain base is sequenced in the sequencing effort. So that is needed for sequencing of both alleles 99% of the time. And in polyploids, that number goes up considerably, as you can see. So that becomes both a bit more complicated technically, but also much more expensive. So And the way in which this is sort of mostly, for example, in the allopolyploids, I guess, when there can be a certain amount of divergence between the two genomes that merged. And for example, if more than 2% divergence is present in the two genomes, or if the tetraploid, for example, so the read mapping can be done with less than 2% mismatches per read, if you set it like that. So that's sort of a theoretical calculation that you can do when you plan your sequencing or genotyping. So that was a little bit about polyploidy, which is another important factor in determining your genotyping platform. And now I'll mention one of the first of the three platforms. They all use next generation technology. And of course, whole genome resequencing, as the name suggests, is that you basically sequence the genomes of a whole panel of genotypes or populations that you are interested in characterizing. And it is especially useful for species where this linkage disequilibrium decays very, very rapidly, such as in maize, olive, perennial ryegrass, red clover, and so on. So many of outbreeding species, that is the case, because then when you do your whole genome resequencing, you in theory get every single nucleotide polymorphism that are there. And so you will have a SNP, the SNPs very close together, and so have a high chance of being in linkage disequilibrium with your trait of interest. And there is a wide range of variation between species in terms of LD. So it can go down to 25 bases in olive, up to 8 megabases in wheat. And I think in the 5 species we're working with, we have a range somewhere in between that. And so price is an important issue there for whole genome resequencing, because it can get quite expensive if you have large panels and high coverage needed. So if you, for example, choose 1x coverage in inbred species, which might be a possibility, and you have a reference genome, so you can impute missing data, and but then you need 15 times or even 30 times in pool populations of tetraploid species such as alfalfa. But if you need high coverage and the genome is large, so the cost of GBS panel, for example, of a few hundred genotypes or samples can be several $100,000. So it can escalate quite rapidly with whole genome resequencing. But it is done more and more in many of the major crop species. So then the next platform, which was used in the species that were talked about in detail yesterday, and Affymetrix and Illumina are the companies that are the main manufacturers of these SNP arrays, and the numbers below there I've taken from the paper quoted. So it's from 2017, so the numbers may have changed slightly, but at that time there was 46 arrays available in crop species, in 25 crop species, and they varied in size from 3,000 markers to up to nearly 1 million markers in certain species. And so these SNP chips, this is a slide I borrowed from my colleague Matt Hegarty, who uses it in undergraduate lectures. And so this is an example of Illumina workflow. You have each chip which are shown there on the left, can do 12 to 24 samples with anything varying from 3,000 to 1 million SNPs per sample. And so the chip surface is covered in DNA probes, which are designed based on known SNPs and the surrounding sequences in that genome. And that you then sample DNA binds to the probes and is used as a template to copy. And then you have the probe goes up right up to where the SNP is, and then you add the SNP. The different bases are labeled with different dyes, and so if the SNP is A or G, A is labeled green and G is labeled red, and then the computer scans and record colors for each of the probes, and so you get green for AA, red for GG, and yellow for AG, which is the heterozygote. So that's sort of very simplified how a SNP chip or SNP array works. So one of the advantages of using a SNP array is it's quite an accurate method of identifying identifying SNPs in your panel of samples, and subsequent bioinformatics is relatively straightforward. Certainly, that's my experience, and there are some disadvantages. As I mentioned, these probes that you use which hybridizes to your DNA samples, they are based on sequences that you already know from the panel you used initially to find your SNPs, and that is just a sort of a finite number of samples. So your SNP array suffers from what calls ascertainment bias, and so you will never discover any novel information that are not present in that initial panel where you identified your SNPs. And you also, as you can deduce, you need knowledge of sequence information. You need the knowledge of where the SNPs are and the surrounding sequence. And you need to genotype many individuals to obtain allele frequencies in highly heterozygous species. So these are some of the pros and cons in SNP arrays. And I'll now go on to say some words about reduced representation sequencing, which was the technology or methodology used here in the eucleg project in the two forage species, alfalfa and red clover. So there's sort of three types, two of which are really important, and they are very closely related. So there's genotyping by sequencing, which was first introduced by Elshire et al. In 2011, and RadSeq, which goes back to 2008, but they're very closely related, as I'll come back to. There is a third method, which I feel I ought to mention, then that's DartSeq, which is based on a sort of a library you make of genome sequences or fragments of your panel, and then you hybridize that to a chip or an array as well. But that suffers from not being very high, what's it called, throughput. So I think there's less and less usage of that compared to the next gen sequencing that I'll go on to now. So genotyping by sequencing is you reduce the amount of sequencing you do by cutting your genome with restriction enzymes, and they recognize very specific small sequences, and you then get some fragments and you sequence a little bit from each of those fragment ends. And then you can detect SNPs, and you do some genotyping simultaneously. So you don't need any prior sequence information. And for genotyping by sequencing, the difference to RAD-seq is, as far as I understand it, is mainly that in RAD-sequence you normally, or in the traditional one, you normally include a size fractionation step, which you don't in the genotyping by sequencing, as published by Elshire. And so it'll also detect presence or absence of restriction sites and small insertions and deletions as well. And so that's all very well, and you can do genotyping by sequencing on individual samples, as has been done in countless of examples. However, when you work with outbreeding forage crops, so you often base your phenotypic data on plots, trials of full SIP or half SIP families or populations, indeed, consisting of genetically different individuals. So they're not inbred samples, as David will explain later in the breeding methodology. So and relating such a plot related data back to genotypic data on individuals is difficult. And so in the eucleg project, the genotyping in red clover and alfalfa is based on genotyping by sequencing technology of pool samples from each of the accessions that we are including in our panel. And so for each accession, leaf samples from 100 or 200 individuals in alfalfa and red clover, respectively, because you don't need as many pool samples in alfalfa because it's tetraploid and you sort of get 2 genomes for the price of 1, whereas red clover is diploid. And so those number of individuals were pooled before DNA extraction. And the GBS technology is used to obtain allele frequency data based on occurrence of each allele in individual SNPs. So so that was the sort of rationale for using pooled data. And so this is sort of a brief description of what we did in red clover. And also with alfalfa, I believe. So you have your genome and you cut with restriction enzymes, and after pilot experiments, the enzyme PST1 and MSE1 were used in both species, and sort of there's a minimum, you want a minimum read count of 30 per certainly in red clover, or I think it was reduced to 27, but never mind. You remove markers with over 80% of missing data, and then you also remove markers having less than 5% informative samples, where such samples are defined as having a reference allele frequency between 5 and 95%. And so yeah, so basically you cut with your restriction enzymes, you add some barcoded adapters so you can multiplex your genotyping effort, and you introduce a second restriction digest, and then you add barcode adapters to those sites, and then you amplify your DNA. So that's very shortly how that was done. I should say that in the case of red clover, Tom Rutting from ILVO developed a bioinformatics pipeline that was used here. So he is really the person who has dealt with the bioinformatics after we had the samples sequenced. So we have a lot to thank him for, and certainly in Red Clover and in Alfalfa, it was the people from INRA, Philip Barr, and Bernadette Julia and colleagues. So for Alfalfa genotype data, which you will hear much more about later, I believe 1,016 samples were collected and then were genotyped. And as I said, 100 plants from each of those accessions were pooled and sequenced. And I think at least 9 million reads per sample were secured. And a number of filtering processes then resulted in over 200,000 markers that are available in the database platform that everybody has used, the Progeno platform that everybody dives into and uses when analyzing data. And so some of the last filters that were used, I got these slides from Bernadette Julier the other day. So you want less than 80% missing values and on the samples and on the positions. So you want at least 10 samples with a frequency of more than at least 5%, and a frequency of minor alleles of 5%, and SNPs with a maximum of 5% missing data. But if you want to know in detail I think it's better to ask the people involved. So for the Red Clover genotype data, we didn't get quite as many markers as they did in Alfalfa. There were 12,251 GBS tag loci identified and were present in over 600 of the 641 samples that were used in Red Clover. And, and those loci were used to identify polymorphic sites and get the SNP allele frequencies from those. And it was found that the average distance between neighboring GBS tag loci was 34 kb, and there were 3.7 SNP markers per GBS tag on average. And so, which is evidence of high sequence diversity, basically 1 SNP per 40 base pairs across the populations. And all in all, over 65,000 SNPs are available in the Progeno database for all our colleagues in the eucleg consortium to work with. And of course, when you do, theoretically, you could genotype each individual, each of the 200 individuals or 100 individuals, whatever species you're talking about, and get very accurate allele frequencies in the population. So when you do this pooled strategy, you do lose a little bit of accuracy, but as you can see here, there is quite good correspondence between allele frequencies as measured in when using individual samples compared to the pooled samples. So I think it's quite well established that it's usable. Then I will go on to I know the 3 of the crops here where we used SNP arrays were already mentioned in some detail yesterday, but I will just do this for the sake of completeness. So there were 400 faba bean accessions genotyped, and with a 50K axiom array with over 34,000 SNPs identified. But the final score matrix included 352 samples with over 22,000 SNPs. and there is some heterozygosity in some of the samples, and initially actually there was a GBS strategy selected for this species, but a SNP array, the SNP array there I mentioned, became available and was considered a superior option, so that was used. For P's, 260 samples were used, and there was a 30,000 SNP array used for genotyping, and over 12,000 markers available for analysis in ProGeno. And also here there are some examples of sort of initial analysis, genomic relationships and genomic predictions of potential RIL performance in P. And finally, soy SNP. I believe I'm running out of time. They used the 355K soy SNP microarray, which was developed by our Chinese colleagues. Over 480 samples, European samples, were used, and 394 Chinese accessions were used. And there's already been published a paper with a detailed analysis of, which is shown there at the bottom, of the genetic diversity in these panels here. So I won't go into that any further. And so I believe this is my last, no, there's one more slide. So these are some of the conclusions that you can possibly take home from this lecture. So when you choose your genotyping strategy, you have to consider whether they're inbreeding or outbreeding crop, the linkage disequilibrium, if it's known, possibly the size of the genome, and whether or not there's a reference genome available, and the purpose for which you want it. The GWAS, you may need a higher density than you do for GS, as was shown in the soybean talk yesterday. And of course, ploidy and budget are important considerations. So I think I better end here before I run over time. Thank you very much, and I'm happy to take questions.
Thank you very much, Leif. That was an excellent talk. We've had quite a lot of questions come up in the questions tab, and we have time to ask a few of those. So I'll pose some to you now. First question, was there a specific reason why you used 100 samples per accession for alfalfa, but 200 for the red clover?
Well, yeah, I think I mentioned it briefly. I mean, you basically, because alfalfa is tetraploid, you sort of get the double number of genomes in 100. You sort of get the same number of diploid genomes in 100 samples as you do for 200 samples in red clover. And I think whether 100 or 200, the exact number, we did discuss that to some degree, but it was decided. I think we have been quite, this is quite a high number. I know of people who are working with fewer numbers when they pool data, but certainly I would say that the higher the number, up to a degree, is the more accurate your allele frequency data will be, everything else being equal.
There's another question concerning the costs, and that the library preparation costs are a major factor, even if you go for a low sequencing method. Do you have any ideas or suggestions on how to better deal with this?
With the high costs, or what?
Yes. The library.
Well, I mean, I suppose you can. We had for red clover, which is the one that I had experience with in this project. We chose to work with LGC, and they made the libraries for us. So all we did was we extracted the DNA, we grew the samples, extracted the DNA, and checked the quality and the amount, and then sent the samples off to LGC, and they did the library. I suppose one could make the libraries oneself, but I wouldn't sort of like to do that. And I suppose you can shop around and get cheaper prices than LGC. I don't know. I don't quite know how to answer it, but
well, perhaps we'll move on to the next question.
Yeah.
But the next question concerns the high rates of missing data points you get with GBS technologies. So do you recommend these for the routine application of genomic selection as a result?
Well, as was sort of alluded to yesterday, especially for genomic selection, you, in theory, it's true that you need an awful lot of SNPs to cover the genome, so that you, in theory, have a genome, have a SNP close to one of your traits of interest. But there is evidence to suggest that you can get away with much fewer SNPs when you do genomic selection, because the genomic relationship matrix that you can use, for example, in GBLUB to calculate your breeding values is also accurate with much fewer SNPs, as was shown by Hilde Muller yesterday with soybean. And I've had similar results when I have looked at data from perennial ryegrass. So I haven't yet checked it out with the red clover, and I don't know the experience of people with who have worked with alfalfa here. But I think certainly for GS, you may be able to get away with much fewer markers. So in a sense, the GBS technology worked well for us in this project.
Thank you. And one final question. All studies with GBS data provide the GC content, which is a quality parameter. But the question is, what does that actually mean?
I must admit, I'm not sure how important it is, but I guess it's to do with sort of the melting point of the DNA. If you have more G's and C's, the higher the melting point. But I don't know whether that's important or not. I must admit, I can't. I don't know. Maybe somebody else can help me.
Is there anybody else who can answer that? Otherwise, we can answer it offline.
Oh, yeah, there's two questions that are basically the same there.
Yeah. Okay, well, thank you very much, Leif. I think we need to move on.
Yeah.
We are running slightly over. So if we now move on to the